This podcast is for informational and educational purposes only and is not to be considered medical advice for any particular patient. Clinicians must rely on their own informed clinical judgments when making recommendations for their patients. Patients in need of medical advice should consult their personal healthcare provider. From UPMC Children's Hospital of Pittsburgh, welcome to That's Pediatrics. I'm Steph Dewar, Vice Chair of Clinical Affairs, Program Director for the Pediatric Residency Program, and a member of Pediatric Hospital Medicine.
And I'm John Williams from the Division of Pediatric Infectious Diseases and Director of the Institute for Infection, Inflammation, and Immunity in Children, or the I4Kids. I am delighted to welcome as our guest today Dr. Laurie Silva. Dr. Silva's lab studies Chikungunya virus, which is a reemerging arbovirus that has recently caused epidemics of febrile arthritis throughout many parts of the world, including, I think, our country. Dr. Silva, welcome.
Thank you very much. Happy to be here.
So let me just start with what you do here and kind of what your role is here. You're a basic scientist, correct?
Yes. I have a PhD in virology, and I just started my lab in July and study Chikungunya virus, or CHIKV for short, because I'll probably mention CHIKV. So my lab is interested in trying to understand the viral and host determinants that contribute to replication and pathogenesis.
So how is it that someone like you comes to a place like this to study something like that?
Yeah, you would not think that I should be in pediatrics studying Chikungunya virus. It does infect children, but it has a similar disease to adults. But it's not particular to children. So I joined the lab of Dr. Terence Dermody in 2009 when he was at Vanderbilt as a postdoc. And he said he wanted to study a new virus, something that was pathogenic. And so we started studying Chikungunya virus together.
And Chikungunya virus, or I like that abbreviation, CHIKV, that's a lot simpler and shorter. What is Chikungunya virus?
CHIKV is a virus that is spread by mosquitoes. It belongs to the alphavirus family, and it's an arbovirus, so it's spread by arthropods. And it has other similar viruses, but it's unique in its own family in that it's only one of 4 viruses. The other are 3 viruses that can be spread by mosquitoes in a human-mosquito-human cycle. So a lot of viruses that are spread by arthropods have a non-human host typically, but these 4 viruses can be amplified in humans and then cause these huge urban epidemics.
So a mosquito can give it to one person, then a new mosquito can bite that person and spread it to another, and it expands. And who gets chikungunya? Where is it?
So it was historically in Africa and Asia, and a lot of people hadn't heard of it. But then in 2004, it reemerged in Africa, and then it spread to all of the islands in the Indian Ocean and then the Pacific. And then in 2013, it spread to the New World, because it was always in the Old World, and infected islands of the Caribbean, South America, Mexico, and even there have been 13 cases in the United States of natural transmission from mosquitoes.
Welcome to America.
So are you looking at ways to prevent this spread, ways to treat this infection, or just trying to understand it?
Yeah, I'm a basic virologist, so I really like understanding how things work, figuring out what's under the hood. I've always been in labs that have a very wide range of how they do science. So from the crystal structures of proteins all the way to animal models. So I'm interested in trying to understand how the virus first attaches to cells to get entry into them. And then once it enters cells, how does it set up shop? How does it set up its replication compartments? What host proteins contribute to that? And trying to understand a little bit how it traffics through the cell. And I'm starting to work on egress as well, how the virus gets out. And then long-term plans are to study some pathogenesis. We just remodeled BSL-3, so that's the other thing about CHIKV. There are different categories of biosafety levels, and CHIKV is a BSL-3. So I run the BSL-3 here at Children's, and we just set up to do an animal model. So we can now do animal experiments. There's a mouse model of disease, and trying to understand what causes pathogenesis.
So it's very powerful, obviously, to be able to study a virus both in cells and in the animal model, because then you can look at it in the context of a whole animal. And you said arthritis. Last time I knew, cells don't get arthritis.
Correct.
But humans do, and I guess mice can.
Yes, they get the swelling. It's hard to tell if they have arthritis. They do have signs of arthritis when you open the joints and look in the joints, but the disease, human disease, is not completely recapitulated in mice.
You don't see them limping around, grimacing in the morning like Steph and I do?
It might be a little challenging to do an arthrocentesis on a little mouse.
That's a small joint, yeah.
Yes.
That would be challenging.
Yeah.
So what are you learning about how the virus, gets into the cells and causes disease?
So that was my first project in Dr. Dermody's lab, was to look at what are called attachment factors. So viruses tend to, they need to bind to cells, and they often have one very specific protein on cells that will allow them, it's kind of like the lock and key. It's the key that will allow them to get into the cell. But those protein molecules are usually low in abundance. And so the virus, in order to sort of surf along the cell, binds to something called an attachment factor. These are often glycans. They can be lipids. Sometimes they're other proteins. Chikungunya virus binds to and attaches to glycosaminoglycans. It's a type of glycan that is found on cell surfaces in high abundance. And there's different types of glycosaminoglycans, heparin sulfate, dermatan sulfate, keratin sulfate, and things like that. So we're trying to understand more precisely about what GAGs CHIKV binds, what are the specific signatures of GAGs that CHIKV needs to bind to, what are the residues on the virus itself that allows it to bind to glycosaminoglycans. Work we did previously identified a single amino acid in the viral binding protein that increases binding to glycosaminoglycans, and that actually led to attenuation in the mouse model. So what we've been trying to do is to find residues that will reduce GAG binding and then see if that actually impacts pathogenesis.
I love that idea of that image of the virus surfing along the cell, looking for its specific little receptor. Does that have anything to do with the changes in the distribution of the virus that you talked about?
So that's one thing we really want to understand is does binding to glycosaminoglycans influence tropism, or where the virus can be found in the body? So as I said, glycosaminoglycans are pretty ubiquitous through the body. There's a huge biosynthetic pathway of more than 20 enzymes that make up different GAGs, so they're very heterogeneous. So we're trying to understand, is it a particular type of GAG, and if it has this certain subunit in, let's say, muscle cells, is that why it's more prone to binding muscle cells and things like that?
And when you do this type of work, do you compare this virus with what we know about other viruses, or is...
So there are a lot of other viruses that use glycosaminoglycans. That's one nice thing about virology, is that a lot of viruses sort of figure out the same tricks to bypass the immune system or to get into cells. And so there are a lot of other viruses that bind to glycosaminoglycans. So I think that's one thing that's unique, is it's really hard to find the binding sites that are important for glycosaminoglycan binding on the virus. And so actually mutating those to get a reduction of glycosaminoglycan is kind of a newer idea to try to understand this important attachment factor.
So if you could, you mentioned before that the one mutant you made was attenuated in a mouse model. That sounds like it could be a candidate vaccine, right? A live attenuated vaccine? Is that possible?
That's actually how it was derived. It was U.S. AMRID. U.S. AMRID was developing a vaccine to chikungunya virus, and so they passaged the virus in cell culture and in mice. And that led to this adaptation to increased heparin sulfate binding, or increased GAG binding. And so it took, that was one of the projects that previous graduate student in Terry's lab, Allison Ashbrook, did, was to try to understand how that mutation affected pathogenesis. And so she changed it back to its more traditional or common amino acid and found that there was attenuation. And so we're kind of envisioning a Goldilocks kind of scenario where too much GAG binding is not good for the virus. It prevents it from spreading well. And then we hypothesize, too little GAG binding is also going to cause attenuation, because we think that the virus won't be able to find its actual proteinaceous receptor if it can't surf along the cell surface as well.
And you mentioned the military. Is that a source of funding, or is that a group that continues to be interested in this research?
No, that vaccine, it's called 18125. It was disbanded because in phase 2 clinical trials, there was some transient arthralgia, and it can revert pretty quickly. So a reversion at that site was found in a patient.
So your studies of understanding what determines this tropism, this binding, could maybe identify another virus that was more of the Goldilocks? Not too hot, not too cold, but just right.
We observed in vitro some strain differences in how these different chikungunya virus strains bind to glycosaminoglycans. So we're wondering if that also influences pathogenesis.
So why is it spreading throughout the Caribbean and to Florida? What's up with that? I mean, everybody wants to go to Florida when they get old to retire, but this virus sounds new. You said it's emerging.
Yes. So like a lot of other arboviruses or viruses that are spread by mosquitoes or ticks or other things, Lyme disease, for example, the world is changing. So climate change is leading to warmer climates where the mosquitoes or other vectors can live longer. Globalization, so a lot of these different epidemics were started by an imported case. So actually the World Cup in Brazil in 2014, I think, is one of the major reasons why chickpea spread so well, because you had people coming from all over the world getting infected while they were in Brazil and then going back home. There have been cases, the other thing that happened during the first epidemic in 2004 was some virus adaptation. So there's two main mosquito species, Aedes aegypti and Aedes albopictus, that chickpea can infect. And there was a evolution and a mutation in one of the strains that allowed it to better infect Aedes albopictus. And that's in more temperate areas like the US. And so there was more cases just because it was able to spread through that mosquito vector.
So we could easily, from both the mosquito vector and weather changes, we could see it spreading north in the U.S.
Yes.
But we'll probably stay for a while here in Pittsburgh, right?
Probably so. But we also are an air-conditioned nation. So very few of us are spending a lot of time outside being bit by mosquitoes. It does happen, but I don't think we're going to see the huge epidemics in the U.S., at least for a while, that we've seen in more tropical areas.
So you mentioned the reason that you found this particular bit of research at Vanderbilt with Dr. Dermody. I'm just curious, why virology from the beginning? How did you end up with that interest or that passion?
I was always interested in science and knew that I wanted to do biology. I was really fortunate to have a really great high school biology teacher who encouraged me to do summer research, and that's kind of when I fell in love with doing research at the bench. When I decided to go to grad school, I wasn't quite sure what I wanted to go to grad school for, and I found a brochure that was basically infectious diseases and thought all of that sounds really cool, because I knew that I wanted to, though I like basic research, I knew I wanted to do something that was disease related. And so I just was looking for programs at the time that had PhD programs in virology. And I don't regret it for a moment. Viruses are amazing. They are tools to teach us about cell biology. They change the dogma that we think we know. They're beautiful structurally. They're simple machines that are capable of replicating themselves without any knowledge to say so. Like, I don't like to anthropomorphize viruses, but they are really cool.
Well, and I have to say, obviously I'm biased as an infectious disease physician, but I think you just highlighted one of the things that's really interesting about infectious diseases and viruses, that they really change over time, right? I mean, we often think in the modern era of diseases going away because of antibiotics or vaccines or window screens. But, we've seen in recent years with people not vaccinating like they should, measles resurging. We've talked about that on this program. And what you just said about, the spread of this virus that 15, 20 years ago, probably few people in the U.S. would have heard of. And now the virus itself is here. So that's pretty amazing.
It's amazing, but on the other hand, it's frightening, right? Because viruses are very crafty. And they survive and they adapt and they change. So, I mean, I find it relieving that there's people like you out there really curious about how are they going about this to understand, their adaptations and their ways to get in.
Yeah, and that's one of the interesting things about chikungunya virus. There are a lot of other viruses like dengue and Zika and other alpha viruses that cause febrile arthritic disease. But chikungunya is one of the few that has prolonged symptoms. So depending on the study and depending on the area, roughly half of the patients have prolonged arthritis or other disorders that plague the patient for weeks or months and sometimes even years. And so really trying to understand what causes this chronic polyarthritis is really important, because it's debilitating when people have it acutely, and it's also debilitating when people have it chronically, and it really affects their quality of life.
Yeah, and that's pretty unusual. There's not that many viruses that cause a chronic, ongoing infection like that, right?
I don't think even before I knew about CHIKV that you could get arthritis from a virus. And it has a lot of characteristics with rheumatoid arthritis.
I mean, we'll see children who have parvovirus, a DNA virus, who will have a transient arthritis, but it lasts a week or two and then it's gone. So it's not at all what you're describing with CHIKV.
CHIKV normally lasts about 3 weeks. I've never met somebody, but it's been described as debilitating during that time. People are on the couch, not moving. Chikungunya comes from the Makonde word, that which bends up, because people have such a stooped posture because they're in so much pain. So that it can last that long seems problematic, to say the least. And it really is a huge economic burden, too, because people can't work. When Laurie was hit, it was about 70% of the people had it, and the hospitals were inundated. Like, basically everything on that island stopped.
Wow. So at least in the U.S., the place that it's first landed is Florida, where everybody's retired.
Yes, and Texas.
Texas, there might be some people still working in
The
Texas.
mosquitoes are big in Texas. So.
Especially after the hurricanes.
Well, it's interesting, though, when you describe the course of the illness, it's debilitating, but it doesn't sound like it's necessarily life-threatening.
No. Only about 1% of people end up dying, but usually those people have comorbidities. So they have some other underlying disease, which the virus, usually they're immune compromised in some way, and the virus just takes over. Because the immune system is actually pretty good at controlling CHIKV. Antibodies will protect people from a secondary infection. It's just that we think that the immune system gets so hyperactive, that's probably what causes the chronic disease.
And you mentioned right now the one vaccine was withdrawn or didn't make it through trials. So right now there's no vaccine and no specific treatment for the virus?
So there's really only supportive care. And then in terms of vaccine, there are a number of vaccines in development. Several have hit phase 3 clinical trials, and there's a couple in phase 2. So it's a great candidate for a vaccine in terms of protection. It's just a matter of who would want that vaccine, unless you're a traveler, which we have vaccines for travelers. But any people in those areas, sometimes vaccines are hard to deliver to those people.
I think what you're saying is the virus right now is in lots of areas of the world where healthcare is more limited, more resource limited.
Yes.
So even if we had a vaccine, it might be a little hard for them to get it.
Yes. Companies are interested in making money from their vaccines.
That would be diseases that occur in wealthier nations with more resources.
Yes. Which, as the virus spreads, perhaps that's going to happen.
Right. You have a different market that's going to be a little more curious about paying for that.
Especially, but all those retired old people in Florida, they've already got arthritis, okay? They don't want more arthritis from CHIKV.
Yeah, and arthritis if you pre-existing arthritis will exacerbate CHIKV. So
okay, there's your market there. The snowbirds, right?
There you have it. Well, we're so happy that you were able to come and chat with us today.
It's great to be here.
I talked more and thought more about chikungunya virus than I ever have in my entire life during this podcast.
Yeah, you'll be a little careful next time you go to Florida, won't you?
I will actually be very careful.
I'm probably going on spring break, and now I'm a little bit nervous.
Wear your bug spray.
Okay. Well, thank you very much for joining us, Dr. Silva. It's been a pleasure.
Thank you.
And you can find other episodes of That's Pediatrics on iTunes, Google Play Music, and YouTube. Be sure to subscribe to keep up with new content. Leave a review and tell us what other topics you'd like our experts to cover. Thanks for listening.